Hello, my name is Scarlett Raine, and in this video, I would like to introduce our work, Point Label Aware Superpixels for multi-species segmentation of underwater imagery. This is work completed in collaboration between the QUT Centre for Robotics and CSIRO's Data61. Coral reefs are some of our planet's most diverse ecosystems. However, they are at risk due to natural and anthropogenic impacts, including climate change and damage caused by tropical cyclones. Marine surveys are used by ecologists to detect changes in the health of coral reefs and to monitor indicators such as species abundance over the long term. Recently, there have been technological advances in the use of semi and fully autonomous underwater vehicles, thereby improving the efficiency and extent of marine surveys. Robotic marine surveys generate extremely large quantities of underwater imagery, which must be analysed. We can automate the analysis of this imagery by using semantic segmentation. Traditional semantic segmentation architectures require training on large quantities of densely labelled imagery. However, it's prohibitively time-consuming and costly for domain experts to manually label coral images for this purpose, due to the complexity of coral images. Instead, our approach uses the large quantities of publicly available photo-quadrat images already labelled by ecologists with randomly placed sparse point labels. Our method reduces the annotation cost and effort required, while still obtaining dense segmentation outputs at inference time. We take the photo-quadrat images and sparse point labels and propagate these point labels using superpixels to generate a dense augmented ground truth. Prior approaches have generated superpixels for this purpose. However, they've only used color features and do not consider the point labels whilst generating the superpixels. Our approach uses deep learned pixel features and the locations of the point labels. We introduce a novel loss function comprised of distortion loss and conflict loss terms to optimize the superpixel centers, such that superpixels encompass single species regions and closely conform to the intricate coral boundaries. The first term in our novel loss function is the distortion loss, which calculates and minimizes the distances between pixel features. This term aims to cluster pixels which have similar deep features and are therefore likely to belong to the same species of coral. The second loss term is called the conflict loss, which penalizes superpixels for containing conflicting class labels. If 2 points have been labeled by the ecologist as belonging to different coral species, then those points should not be contained in the same superpixel region and should therefore have dissimilar superpixel memberships. We optimize the superpixel centers, convert pixel soft memberships to hard memberships, and propagate the class of the member point labels to every pixel inside each region to obtain a dense pixel-wise augmented ground truth mask for each image. For the label propagation task, we outperform the current state-of-the-art CoralSeg by 3.6% for pixel accuracy and by 8.35% for mean intersection over union, when the augmented ground truth masks are evaluated on the UCSD Mosaics dataset. We also reduce the computation time per image to just 1.37 seconds, which is an improvement of 76% over the prior fastest approach. The example on the slide demonstrates that only our approach is capable of capturing both the green and beige coral segments in this example. This is a visualization of the superpixel regions as our point label aware loss function is minimized. In this example, we can see that the superpixels are optimized to form single species coral regions. The superpixels closely conform to the intricate boundaries of the coral, creating an accurate augmented ground truth. At each iteration, we can see in this example that the superpixel regions change in the image as the cluster centers are optimized. If we look at the superpixel segments in the top right image, we notice that they spread out where there is only one class present, such as in the top third of the image, and the superpixels cluster closer together and form smaller regions in the more complex parts of the image, such as in the middle section of the frame. We use our novel point label aware superpixel method to obtain dense augmented ground truth masks for training images. These dense masks can then be used to train any semantic segmentation model. In our work, we used the DeepLab version 3+ architecture and trained the model on masks obtained using our novel approach to superpixels. This model can then be used to perform inference on new coral images. We evaluated the segmentation performance on 2 publicly available coral datasets, the UCSD Mosaics dataset and also the ILAT fluorescence dataset. We outperformed the state of the art for the semantic segmentation task on both of these datasets. Specifically for UCSD, we improved on pixel accuracy by 2.9% and mean intersection over union by 9.7%. For the ILAT dataset, we see an improvement of 4.2% for pixel accuracy and 14.3% for mean intersection over union. Our method leverages ecologist labeled points in coral images and facilitates fast annotation of new images to allow training and retraining models for new locations, conditions, species present, and based on the interests of ecologists. As new species are encountered or as ecological priorities change, the model can learn about these classes without densely labeling the new images manually. In summary, we have contributed a novel loss function comprised of 2 terms and have demonstrated improved performance on both the label propagation and segmentation tasks. Furthermore, we make our source code publicly available on GitHub. Thank you for watching our video.